Yeah. Okay, well, good morning, everybody. I'm Ali Fatemi. I'm a pediatric neurologist from Kennedy Krieger Institute. And this is Dr. Manu Amanat, also from Kennedy Krieger Institute. He's a postdoctoral fellow. And we are tasked to provide a brief summary of the scientific meeting, which we had this year on Thursday. It was a great meeting. We had the speakers talking about various leukodystrophies, and it was mostly focused on new discoveries in terms of clinical discoveries and scientific discoveries related to known syndromes or to known diseases. So in general, I think this is important to point out, is that really over the last sort of 10 years, there has been dramatic progress, even though as an individual patient or family, you may not feel it. But we now have several leukodystrophies for which there's clinical trials. There's different therapeutic trial approaches. 10 years ago, most of these approaches were, daily pills or molecules, small molecules that one would take to try to sort of stop a process. Now we're talking about gene-targeted therapies often. So we either talk about gene therapy, things that actually bring in the missing gene into the cell, or different approaches like ASOs. So these are small molecules called antisense oligonucleotides that go and can block a gene or can block a specific mutation of a gene. So some of these speakers are also presenting today. So this is intended to be really just a short summary. We're not going to get into the details. Specifically, we sort of tried to alphabetically sort it, but it doesn't look like it's alphabetical. But anyway, so actually, right after us is Dr. Hashemi, who's presenting. She's a member of Dr. Van Haren's lab in Stanford, and they presented a new model for adrenoleukodystrophy, taking the ALD mouse and basically making it more sick. And she'll talk about it herself, so I won't get into details about that. The only thing I wanted to mention about that is that there, outside the scientific meeting here, there has been a lot of progress in ALD. Most importantly, there was recently, just a couple of weeks ago, an FDA advisory board meeting that voted unanimously yes to approve gene therapy, ex vivo gene therapy for ALD. That doesn't mean that it's going to get approved by the FDA. They decide that themselves, but we are excited that at least the expert committee agreed that this is something that should move forward. So that's, I think, the most important progress there. And then you want to talk about Alexander?
Hi, everyone. What I want to talk about is the Alexander disease. So for this disorder, Dr. Tonduti talked about a classification that he proposed to actually classify patients based on their evolution of their disease. So we have different types of categories based on the neuroradiological features, and one of the most recent ones is just classify patients based on their clinical symptoms and their imaging features, and we have it in type 1 and type 2 categorization. And what these type of classification doesn't describe about the evolution of the disease and the future disease trajectories that are important for the clinical trial readiness, and we could actually stratify the clinical trials based on the evolution of patients. So Dr. Tonduti proposed a modified version of the classification that based on that the type 1 ALD, Alexander disease people have 4 other subcategories. And based on that, we could actually have patients with different types of evolution. And then also we have the type 2. So this is important because the only drug that is ready to be used in the clinical trials, phase 1, phase 2 clinical trials, is a molecule called antisense oligonucleotide. And we need to know about the evolution of patients, the future disease trajectory in patients, so we could actually have a better and effective type of clinical trials. So the next one is AGS.
So yeah, the next speaker was on Leukodystrophy syndrome. Dr. McKenzie from University of Rome has been doing some really innovative work. So Leukodystrophy syndrome is basically a group of diseases that is due to genetic changes in genes that are involved in intracellular response to inflammation. And so there is an altered response, and it's usually related to changes in the way RNA is processed. So just a brief, what is RNA, DNA? So, you know, each cell in our body has 3 billion DNA letters inside the center of the cell, called the cell nucleus. And so the DNA is basically the genetic information. It's like the cookbook, right? It's the actual book, and it has many chapters. We call them each like exons and so on. And that information from that cookbook, in order to be transcribed into an actual information that goes outside of the center of the cell, that DNA is copied into something called RNA. And so that RNA is a single strand molecule that leaves the center of the cell, goes to the cytoplasm, to the other areas of the cell, where then that information is used to make the proteins. So it's basically a photocopy of the DNA. But it also has other functions that we're just learning. And so in one particular form of AGS, one of the genes that is involved is called ADAR1, which is a so-called RNA editor. And so what it does is it basically can change the individual nucleotides here for the RNA. And so it's mutated. And when it's mutated, that RNA editing doesn't work properly. And as a consequence, a whole host of genes, when they are being transcribed and they're supposed to be fine-tuned, that fine-tuning doesn't happen. And so he proposed something called site-targeted RNA editing, which is a molecule that can go in and basically correct that mutation, not at the gene level, so not editing the DNA itself, which is what, for instance, CRISPR does, but at the RNA level. So if this is the DNA at the center of the cell, and that's transcribed into RNA, that RNA comes out, and you can edit the RNA here rather than editing the gene itself. So this is a very interesting approach that he's pursuing right now, and he basically just made that proposal and showed some data in the preclinical model that he has. So next was on a group of diseases called microgliopathies.
So to talk about the microgliopathies, so let's first talk about the microglia. So we have an immune, when we talk about the immune system, we have a cells called macrophages. And these macrophages, when they are not in the blood, we call them macrophage. But when they get into the different tissues, they get different names. For example, when they're in the liver, we call them Kupffer cells. Or here, when they use a specific type of receptor, or the CSF1 receptor, they get into the CNS, and we call them microglia. So the mutation in the gene that actually make this CSF1 receptor, which can be a gain of toxic function or loss of function, what will happen is that we're not having the proper microglia in the CNS, and this can cause a disease called ALSP, adult onset leukoencephalopathy, with axonal spheroid and pigmented glia. So this disease is, when we look at the pathology of ALSP, it's like other diseases like ALD. And for example, in there, we also have some problems in the microglia. And we know that for ALD, we actually use the most approved type of treatment is hematopoietic stem cell transplantation. And what Dr. Bergner actually proposed was that because we have the same pathologies, maybe the treatment that we could use, for the ALD can also be effective for this ALSP2. And the hypothesis here is that this microglia can actually move into the CNS when we do the hematopoietic stem cell transplant. They have these microglia, these stem cells, and these macrophages could actually go from the peripheral tissue and blood to the CNS and make the corrected and has the corrected macroglia. So it should be noted that the hematopoietic stem cell transplantation has its own adverse events. For example, we could have graft versus host disease. So the idea is that when these macrophages, these new immune cells go into the cell, they could actually attack the human cell body. That we call them as a graft versus host disease. So we need to have, there should be other studies to actually assess the safety and also the efficacy. But this is the hypothesis to use this type of treatment for the ALSP2. The next one is about the MLD.
Yeah. Just wanted to make one more point on ALSP that Dr. Bergner highlighted, was that when she looked at the brain sections, autopsy material of patients who have died of ALSP, and she compared that to patients who have died from ALD, she saw overlapping features. And that's, I think, what Manu was trying to say. So there is an area where, next to the lesion, where we actually see loss of these microglial cells, which was actually an observation made by Florian Eichler about 15 years ago in ALD. And it wasn't clear why is it happening. And so now we know that this is sort of when these microglia get sick, there's a dysfunction, and so they can also be lost. It's not just that they become aggressive, they can also become, they can die, basically. But both of it is bad. So whether you have too much of an inflammatory cell or too little seems to be bad. The other feature that she could see in between the two diseases is what we call these axonal spheroids. So basically abnormal cables, nerve cables that are being cut off, and they swell up as a consequence. And so that's interesting, because there's this overlap. And then most recently, there has been another syndrome due to mutations in a gene called AARS2 that clinically can look a lot like ALSP, and pathologically can look like it, but it's a completely different type of mechanism in terms of the gene. And so the question that basically Dr. Bergner raised at the end was like, how many diseases are out there that we could potentially use this bone marrow transplantation process to replace this sick microglia with healthy ones from other people? And so I think that's a still open question, and I anticipate more data will come on as people are getting transplanted across the world. So there's a series of patients that have received bone marrow transplantation with ALSP already. There's some data that's been published from Europe, and also in the US, a series of patients been transplanted in Boston and at the University of Minnesota. And we're still learning, but it's an important progress. The next speaker was Dr. Hooper, who's just here in the room. He talked about metachromatic leukodystrophy animal model, focused again on the mouse model that's been out there for a while, but there were some new things that he reported. One of the interesting things that he reported was, that this is a disease that affects not just the brain, but also the peripheral nerves. So in metachromatic leukodystrophy, peripheral neuropathy is often a very prominent feature and a very disabling feature of the disease. And you can take these mice's Schwann cells, which are the cells that produce myelin in the periphery. So there's two types of myelin producing cells, the ones in the brain and the central nervous system, and the ones in the peripheral nervous system. And the ones in the peripheral nervous system, we call them Schwann cells. And he cultured these, and he saw that they are releasing pro-inflammatory markers, call them TNF-alpha and other things, which is really interesting, because this basically may imply that there is a pathological processes happening that are immune-mediated, outside the central nervous system as well. The next speaker was from, I think he's talking later today anyway, but maybe a brief summary about it.
I talked about the microglia, right? The central nervous system has different cells, neurons, microglia, and the other one is astrocytes. So what astrocytes do is that for example, it makes blood-brain barrier, and it's also connected to different neurons, and could remove the excess, for example, potassium in there that could help to avoid the cerebral edema. So what Dr. Min was talking and explaining is that all these projections that you see from the astrocytes, they're actually in the connection with the neurons. And so these astrocytes get all the excess ions that could be actually toxic for the neurons, get them, and from the input, it could just dump all these ions to the blood. And the role of the astrocytes is now really important. And we have a disease called megalencephalic leukoencephalopathy, or MLC, and we found that most of the patients with this disease have mutations in two genes that encode MLC1 as a receptor, and also GliaCAM1. Yeah, and these two receptors are connected with other types of receptors that could actually regulate the removal of potassium, chloride, and all other different ions. This is now important that the mutation in so this is all about the underlying mechanisms that could, cause to understand about the, treatment of the disorder in future. So when we see that there is a mutation in these genes, we could see that they indirectly also affect other receptors. And what it can happen is that we see cerebral edema in the CNS that could actually cause all the symptoms of the patients. And with targeting these receptors in future, we may actually have some treatments for them.
I'll say a couple of things just to correct you. It's not receptors. These are proteins, okay, that are mutated. So they're proteins that connect the membrane of the cell with what we call intercellular space. Okay, so there's this layer here in our blood cells where blood is flowing inside the vessels, right? And here's the brain. And so there's something that you mentioned briefly called blood-brain barrier. The blood-brain barrier is very important. It's something that regulates what can get into the brain and what can get out of the brain. Okay, it's extremely tightly regulated, and so otherwise it will be a disaster, because our blood, when we eat food, sugar levels go super high. When we eat fat levels go high. The brain makes sure that everything is always tightly regulated, what comes in and out, and that's done by these astrocytes. And so these two proteins are key proteins that are involved in making sure that this connection between the astrocyte and the blood vessels are intact, right? So this is the intracellular space and the membrane of the astrocytes. So they're not receptors, they're just proteins, but they interact with a bunch of other proteins and the things that transport things in and out. And so the remarkable thing is that these closely interact with each other. And that's why you can have this disease where you have a mutation in this gene, the MLC1, or whether you have a mutation in this gene, GliaCAM, because they're so interacting with each other. So this is a common theme in biology, is that when we see that genes that are related to each other, they tend to often cause a very similar, if not the same disease, because it's the same mechanism, sort of the same pathway that's involved. And we'll get to that in a little bit with some other diseases. So in vanishing white matter disease, we had a lot of actually focus on it during this meeting. there has a lot that has happened on vanishing white matter disease in the last few years. There is a consortium now, an international consortium that's headed by Dr. Van de Knaap in Amsterdam, that includes several academic centers, trying to come up with what we call clinical trial readiness. So clinical trial readiness means, all these discoveries in the lab, that's fantastic, right? But how do we actually make sure that we get into the patients, the new drugs, and when we get new drugs into the patients, that we ensure that we are actually having a solid way to measure the effect of the drug. And so part of the research has to be to exactly understanding the disease course. And so the first speaker was Dr. Van de Knaap herself, who focused on that. So she has a very large database at the University of Amsterdam now, data that has been collected for several decades now on patients with vanishing white matter disease. And she's been digging deep into trying to understand all the different characteristics of patients that may define how they will progress. And so one of the, for instance, important things that she's found out is that the earlier the onset of the disease, the more rapid and the more severe it is. And she can basically categorize patients in different groups based on age of onset. So this is critical as we are now finally moving towards clinical trials. And I can't tell you exactly who these companies are, but there is at least 2, 3, maybe 4 companies out there that have a drug that may target vanishing white matter disease gene mutation. But we need to decide, okay, which patients are we going to go after? Which groups are the best ones? So these are things that we've been talking about based on this data. And she described some new MRI findings that were also not previously known that may again help in stratification and also guide us as to what to look at. So one of the things, for instance, that looks like to be the case right now is that we can't really use MRI as an outcome for this disease because the MRI doesn't necessarily always correlate with the clinical course. And that also the MRI can be quite different. In younger patients, there is what we call the vanishing of the white matter. In patients with later onset disease, the white matter doesn't necessarily vanish at all. It may not look like vanishing white matter disease, but they may instead have more atrophy, shrinking of the brain. And so there are these different things that we are uncovering. There's now changes that we see in the brainstem, and that seems to be a different pathology, a different type of cells involved. And so these are all important discoveries that are being made right now. So then her presentation was followed by Dr. Hung-Yu Shi. Hung-Yu Shi is a neuroscientist in the lab of Dr. Josh Bonkowski in Utah, and they have a zebrafish model for vanishing white matter disease. So zebrafish are great for making animal models. You could technically eat them, but I don't think that's happening in the labs. But what's happening is that they grow really fast. And so you're talking about zebrafish embryos, basically. And when things replicate fast, beings replicate fast, it gives you also an opportunity to cause mutations in a quick pattern. So we can cause pretty easily mutations, induce mutations into the genes of zebrafish. And even though they're fish and not human, they may sometimes develop the same or similar features. And so you can cause mutations in the vanishing white matter disease genes in the zebrafish. And then you see that the fish don't swim correctly and they have changes in their brains, in their central nervous system structure. And one of the things that we know is involved in this disease is what we call the integrated stress response. And so that's really critical because all these new drugs that are right now out there are trying to focus the integrated and basically block the integrated stress response in vanishing white matter disease, because the gene itself that's mutated, or the genes that are mutated, it's actually a complex of 5 different genes. They are the master regulator of the stress response, and this integrated stress response is something that we all have, and it's an evolutionary important thing that our cells need to do to protect themselves against stress. But when it's out of control, and when the stress response is too active, then you get sick. And so the drugs that are being used are called integrated stress response inhibitors. And Dr. Shi showed that in the zebrafish model, not only be a clear signature of integrated stress response in the vanishing white matter disease zebrafish, But this stress response is mediated through something that we call stress granules. And so these stress granules have these little granules inside the cells that contain the markers of integrated stress response. And they went on and showed that if they knock out another gene in the vanishing white matter disease gene called STAUFN1, they can improve the integrated stress response. So basically, they're showing that if you can block a gene called STAUFN1 in vanishing white matter disease, you may get improvement. Now, of course, the question is, are the drugs potentially out there that could block the STAUFN gene? Because you can't in patients knock out the gene. You can only do that in zebrafish, but that seems to be the case. So it's a potentially new therapeutic avenue for vanishing white matter disease. The last speaker on vanishing white matter disease was Dr. Piscopo from Canada, and he has taken patient cells with vanishing white matter disease and turned them into stem cells, and then differentiates those stem cells into glial cells, so astrocytes and oligodendrocytes, the cells that you already heard about here. And one of the things that he seems to be observing here is that the process of these oligodendrocytes, the myelin-producing cells, is slowed down in vanishing white matter disease. So those cells, they are coming from so-called oligodendrocyte progenitor cells that then become mature cells and make the myelin, and that process seems to be slowed in these cell lines. So it was a lot of new discoveries and interesting findings in vanishing white matter. And then lastly, there were 3 speakers on hypomyelinating disorders.
So hypomyelinating disorder is another type of leukodystrophy, and we have different types of diseases in there, like PMD and HCC, that which is hypermyelination with congenital cataract, and the other ones that I will talk about them. So Dr. Wolf, she talked about, so this is interesting that since when we were in 2011, over half of the patients had unknown genetic mutation in the hypomyelinating disorder, and we didn't know about the type of mutation that they had. And then with genetic studies and whole Sanger sequencing, whole genetic sequence, we got the genes that are actually involved in these diseases. So Dr. Wolf talked about different genes that could be actually involved in this disease. For instance, she talked about the proteins that are responsible to make the lipid membrane. And this is important because when we say that this protein is making the lipid membrane, it could actually have other symptoms too. For instance, so here you could see that this protein is FAM126A, and this protein is involved in making the disease called HCC, or as I mentioned, hypomyelination with congenital cataract. So Dr. Wolf mentioned that mutation in this gene not only make the hypomyelinating disorder, but also can cause other diseases, for example, inflammatory bowel disease, or even types of cancers like non-Hodgkin lymphoma that were reported in rare cases. Also, we have other types of, so again, as you could see, we know that FAM126A could cause the disease HCC, but also can, all these proteins that are in connection with FAM126A can also cause the disease, and it is important to know. We also have other types of genes that Dr. Wolf talked about them. For example, we have TMEM106B. It could cause another rare hypomyelinating disorder, but again, because it's important in making transmembrane protein, it can cause other diseases, systemic disorders like frontotemporal dementia. We also have a new category in the hypomyelinating disorder, because hypomyelinating disorders are mostly permanent diseases without any remyelination or repair. But we also have, and Dr. Wolf actually reported the cases with transient hypomyelination. For example, we have TMEM63A mutation, and patients with this type of mutation have a transient hypomyelination, but after a while, this hypomyelination can be repaired. And it is important to know the mechanism. Unfortunately, right now, we have no idea why this is happening, but this is important to know the underlying mechanism to actually understand the possible way to cause the remyelination and repairing of the myelin. So the next speaker was Dr. Sunatra Sais from CHOPS, and so she talked about hypomyelination with atrophy of the basal ganglia and cerebellum, or what we call as HABC. So in this disease, we have most of the patients have the mutation in a gene called TOP4A, and she actually developed a mouse model of this disease. So what we should know is that in the TOP4A is important to make beta tubulin, and beta tubulin in the connection with alpha tubulin is important to make microtubules. So microtubules is a protein that is making the, for the movements of different molecules in the cells, we need the microtubules, and the defective microtubules could cause different symptoms. So she developed a mouse model with this type of mutation, which is a gain of toxic function. And she realized that the mouse model have the symptoms that are like the human patients with this disease. For example, the tremor or the different types of movement conditions that the patients have. And then what I want to talk about again is about a type of molecule called antisense oligonucleotide. And we can use this type of molecule to target these genes like the TOP4A to actually downregulate the toxic gene. So what she did, she developed a type of antisense oligonucleotide that could target and reduce the transcription and translation of TOP4A gene, and she actually gave the different doses of this ASO to the mice. So this is very preliminary data, because although ASO, antisense oligonucleotide, were developed for different other disorders, we still don't know about the toxicity of it in many of the diseases, including this one. So she's already working on different doses to understand the safety of the antisense oligonucleotide for HABC disease, and also to understand if they are actually effective. And the last speaker was Dr. Stephanie Perrier. And she talked about other rare type of hypomyelinating disorder called hypermyelination with hypogonadotropic hypogonadism and hypodontia, or 4H disease. So a very general mutation that we have in these patients is mutation in POLR3A gene, and she was talking about the severe cases with this condition, because only a few cases have been reported, and it's very important to know about the clinical symptoms of these cases and the evolution of the disease for these patients with severe phenotype. So she talked about 6 cases, and what she mentioned was about the brain involvement of the patients, that, for example, they have involvement and atrophy of the thalami or the basal ganglia. So these structures in the brain, they are important mostly for the movement and also for the sensation. And it is also important to know that, so when we have these severe phenotypes, it's also important to know about the connection of these phenotypes to the genotypes of the patient. So they're right now working also on the genotypes of the cases to understand if there is a correlation between this severe phenotype of the disease with their mutation.
So basically, this was the day. So one thing I wanted to come back to is a few things that maybe were a little too complicated that we try to explain right now, so I just want to reverse that. So when we talk about gene mutations, there is different types of gene mutations, and imagine a gene being the recipe of a cookie, okay? And so let's say it's a recipe that tells you how to make a chocolate chip cookie, right? You can have a type of mutation or an error in that recipe that results in no cookie. That's what we call a loss of function. So you just don't get a cookie, or you get less cookie because you said take too little flour, right? You could also have a mutation or a misspelling in that recipe where instead of adding 10 grams of sugar, it says add 10 grams of salt. Now you have a cookie, but it tastes nasty, right? So that would be what we call a gain of function mutation or a toxic mutation. So that's important to distinguish, because when you want to try to treat these diseases, if you have something where, like for instance in ALD, all the mutations we know so far are loss of function mutations. The patients are missing that product. They're missing the cookie. So all you need to do is figure out how to get that cookie into the cell. And so that's what we use gene therapy for, right? But on the other hand, in a disease like Manu just mentioned, that what Dr. Sassi is doing is she's doing this ASO approach, which is also the approach in Alexander disease. There we have these mutations that are called gain of function mutations. So there are mutations that still make a protein, but that protein is not doing what it's supposed to do, and it's doing something else. And so if we just bring in the regular protein, the regular gene in there, that's not going to help the patients, because they still have this toxic, the nasty tasting cooking in there that they can't get rid of. So what the ASO helps with is the ASO goes in, sees where that mutation is, and blocks it in a way so that you don't make that abnormal product. You just make the normal product. You sort of block that, that 10 gram that is becoming 100, it blocks that 0 in a way. It's more complicated, of course, than a cookie, but that's sort of the fundamental idea. And so that's why we have these different approaches. We talk about the ASOs for these gain of function mutations, and then we talk about gene replacement therapies for loss of function mutations. And the most recent things that are happening in the field, that you've heard about all probably, is this CRISPR approach, where we call it gene editing. So here, you're actually trying to go and correct that DNA sequence within the cells. The challenge is that to deliver those constructs that are needed, those basically workers that are needed to go in and do that, is quite complicated, because they're big and they don't fit into the regular things that we can use to get things into the cell. It's also that they may go and edit other genes that we don't want to do. So that's what we call off-target effects. And so, but it's evolving. I mean, this is happening now. There's some ex vivo gene therapy now happening already, where people go in and try to edit the gene. And we didn't have a presentation about that this year, but this is stuff that I anticipate to hear in the coming years. So this was a summary, and again, some of these speakers are talking again today, I'm gonna, I think, transition over to Dr. Van Haren and Hashemi, who are going to give a review of the ALD animal model. Thank you.